Okay, so we are going to talk about some defenses of the innate immune response. And these are going to be constitutive, which means that they are always present. They're just not on until they get activated in some way. And one of these constitutive defenses that is important and well characterized is the complement system. So we'll talk about complement. So if you have an infection is only going to occur if some organism pathogen breaks through physical barriers. So we have our physical barriers. Are penetrated. And then your physical barriers, such as your skin, your pH cilia in your respiratory tract, etc., pH in your stomach, those do a good job of keeping things out, but if they get past, then you can get an infection. Next up, after the pathogen gets through those physical barriers, are going to be molecular barriers. And molecular barriers are going to be some constitutive proteins that are present and can function immediately when a pathogen does break those physical barriers. So after you break through the physical barriers, you basically engage on molecular barriers, and some of them are going to be discussed here, not all, but we're going to focus on things like complement, interferon, acute phase response, and things like that. So here's a laundry list of differences between the innate and adaptive immune response. We are going to focus right now on the innate response, so keep in mind, so the innate response's key roles are going to be, 1, to use patterns, those pattern recognition receptors, so patterns on pathogens, to recognize those pathogen-associated molecular patterns, or PAMPs. And this is to enable the innate cells to respond rapidly to many pathogens, not specifically. And then the second is the innate immune system needs to alarm the acquired immune response. So it's the alarm system. And it does this alarm through secretion of proteins such as cytokines and chemokines. Now remember, cytokines are there to sort of activate, and sometimes they form an address where the cells need to go, such as IL-8. Chemokines are going to give an address. So that's their main role, is to say there is an infection in your gut, or there is an infection in your foot, or there is an infection in your liver. So the chemokines will help guide your acquired immune response and other components of your immune response to the site of infection. But in general, as we've been discussing, there are some key differences between the innate and adaptive immune response. Most of those are going to be centered around how your different components of your immune system identify pathogens. So remember, adaptive or acquired are key to see self versus non-self. They're going to use antigen recognition molecules. They are going to have immunoglobulin that bind to specific antigen structures. They're going to have a T cell receptor that has to bind to a peptide, so only protein antigens on an MHC. Whereas on the other side, your innate is more focused on, is this really dangerous, or am I just clearing out dead or dying cells? So am I looking at a danger signal, or am I looking at something that I just need to clean up to maintain homeostasis? Now, your innate is not going to be specific. It's going to use pattern recognition receptors to identify pathogen-associated molecular patterns for those PAMPs. They are going to have no memory, and very important is they have a limited capacity. They only have about 100 different receptors, whereas they have about 10 to the 18th different receptor possibilities on the adaptive or acquired side. Now, other differences are going to be that you have constitutive components and the response is in minutes. And here, the response is going to require genetic events. So it's not constitutive, it's not ready to go. And it's going to require cell growth. It's going to take days to weeks to respond initially. And then you get a memory response. Of course, that would be much faster. Now, with respect to our genetics, we do have germline genes involved in our innate as well. Those are our C-reactive proteins, toll-like receptors, surfactants, etc. You also have differences between memory and no memory. So we have memory and specificity on the side of our adaptive, no memory, no specificity on the side of our innate. And another key point is that your innate is really not responsible for autoimmune diseases. It really is your acquired immune response that is responsible for those autoimmune diseases. And so just keep that in mind. We do have hypervariable regions and genetic recombination in our adaptive that we do not see in our innate response. Our innate response, again, only uses patterns, toll-like receptors, flagella, doesn't matter if it's on an E. coli or salmonella, whereas our adaptive, if it sees a flagella antigen, it's going to be specific for the type of antigen. So E. coli have different bacteria have different flagella antigens, so our adaptive response would be able to tell the difference. Our innate response would not be able to tell the difference. So when we talk about barriers, we are talking about several layers that are going to protect us before the pathogen even gets in, before our alarm system goes off. And so we have our dead keratinocytes that colonize the surface of our skin. So these guys are at the surface. And they are going to prevent penetration of organisms. Of course, this relies on intact skin. So any damage to the skin can, of course, cause this barrier to fail. The living keratinocytes are part of the innate system. And these guys are going to be able, if they are damaged, to secrete IL-8 to get those neutrophils to come. They can also secrete tumor necrosis factor. One of our acute phase response cytokines. And so this is going to induce inflammation. Now we also have a specialized cell of the skin called the Langerhans cell, and this is a dendritic cell. It was one of the first dendritic cells ever discovered, and that it has beautiful dendrites, and it was sort of the canonical dendritic cell for a very long time. But it is a specialized skin dendritic cells. And these dendritic cells will migrate to secondary lymphoid tissue, just like any dendritic cell would, and activate your T cell. Nothing different here. Now your skin can respond rapidly to stimulation, and this allows it to activate and inform the adaptive immune response when there is a pathogen that does enter and the innate response is unable to get under control. Now there are barriers in the respiratory tract. We have an upper respiratory tract and a lower respiratory tract, and so the upper is considered the nose to the bronchioles. And in this area of your respiratory tract, it relies heavily on mucus, and mucus is going to be secreted by your goblet cells. And then your mucus is going to interact with the cilia that are on your epithelial cells, and the cilia and the mucus is going to help push the organisms out via sneezing and coughing. Now, one thing to keep in mind is that the secretion of mucus in your upper respiratory tract is dependent on IL-4. So that's going to trigger mucus. And the mucus secretion by the goblet cells is abnormal in cystic fibrosis patients. And then you have your cilia, which are important for pushing things out, is going to be defective in ciliary dyskinesia. Now, in the lower respiratory tract, you don't want mucus and cilia because these are going to obstruct the gas exchange, CO2 and O2. So in the lower respiratory tract, we're going to have surfactant, and surfactant is going to be made by our type 2 pneumonocytes. I think I spelled it wrong. Pneumono. And surfactant is basically proteins and phospholipids, which are important for keeping your alveoli from collapsing during expiration. But surfactant also contains collectins. So let me write this here. So surfactants are important for keeping your alveoli from collapsing. During exhalation. And they have collectins. So some of those proteins are in surfactant are collectins, and collectins are going to have the picture shown over here, and collectins have a globular head. And this is going to bind to sugars on microbes. And it has a tail. And the tail that is going to bind to some receptor on a phagocytic cell. So here you see you have a collectin that's bound to bacteria on the globular head, and then the tail will bind to the macrophage, and the macrophage will eat the collectin as well as the bacteria bound to it. There is a specific collectin called mannan-binding lectin, or, oops, I don't want that color, MBL, and this is part of the complement system. And so this is going to activate the complement cascade. Let me do this. Oops, sorry. Here's my complement symbol. Okay. And so just to do a side note about the gut, because your book does talk about the gut. The gut, of course, in the stomach has low pH. And most pathogens cannot pass. Your gram-negative bacteria and your capsid viruses can pass. And when we talk about these pathogens, we'll understand why. But most things, when it gets to the gut pH or the stomach pH will definitely neutralize. Another thing is your gut is colonized. Your small intestine plus large intestine are colonized with commensals. So commensals do many things. Your commensals are typically harmless because they're outside your body. If there is a break in your gut epithelial layer and they do gain access, they of course can become antigenic and can cause inflammation. But because they're outside of your body, they don't produce toxins. They don't damage your gut epithelial layers specifically by those release of those toxins. They just kind of hang out, and they do many things. They can be in part of the bilirubin digestion or breakdown of the bilirubin. They are also going to digest fiber for us. So they have the ability to digest fiber and helps us feel full. They will also, if we eat too many sugars, they'll use sugar too. So they'll break down those sugars. And they take up space. They take up space, so your more harmful bacteria that potentially can secrete toxins and damage your gut don't grow. Your gut also, epithelial cells will secrete TGF beta, and that of course is transformation growth factor beta, and this triggers the inducible Tregs. It triggers oral tolerance, which is important because you don't want to respond to food proteins, for example. Now, there is another molecule called interferon. Interferon basically was named this because it tended to interfere with virus replication. So interferons are produced by many cells. Interferon 1, I should say. And interferon 1 is considered alpha beta. There are different Greek letters. They have many interferons, but typically alpha and beta are the most potent. So interferon 1 is produced by many cells. And a very specialized dendritic cell is a major producer of interferons during a virus infection. So we have our plasmacytoid dendritic cell. It's a major producer. So it secretes interferon in response to toll-like receptor 3 engagement, which is double-stranded RNA. And of course, we do not have double-stranded RNA in our bodies, and so that triggers the plasmacytoid dendritic cell to say there is something else happening in us. Now, interferon prevents virus spread, and it can do this through preventing translation. So basically, a virus will hijack your cell, will take over your translation machinery, so it makes proteins for the virus, not proteins for the cell. And so interferon is going to interact with our translation machinery and shut it down. So no virus proteins will be made, but also no cellular proteins will be made. And so it does have that side effect of inhibiting even protein synthesis on this, or the cells. So the cells, if the virus doesn't get neutralized, well, they could potentially die too from that. So it can also be diffused and act in a paracrine manner. It's going to activate natural killer cells and antigen-presenting cells. So for the antigen-presenting cells, it's going to stimulate TAP, transporter for antigen presentation, and it's going to increase MHC1 proteasome. So that's the proteasome that's going to make sure that the peptides are the length required to load on MHC1. It also promotes a Th1, because remember these are intracellular pathogens, and so you want a Th1. Recombinant interferon alpha was used to treat hepatitis C virus infections. And this was, the R stands for recombinant because it was made in a lab. And it can also be used for treatment of some cancers such as chronic myeloid leukemia. And with chronic myeloid leukemia, the gene BCR-ABL is involved. With the success of that treatment. Okay, so the next thing to talk about is the acute phase response. We have talked about the acute phase response. This is going to be triggered by the release of three main cytokines, IL-1, IL-6, and TNF. The proteins are going to increase what's called the erythrocyte sedimentation rate. Which is going to be diagnostic. So to see if someone's having an acute phase response, you don't measure IL-1, IL-6, or TNF-alpha. You can measure their ESR, which is used as an easy way to measure whether or not someone is undergoing inflammation. The acute phase proteins are going to affect the hypothalamus. And they're going to increase the body temp. And this is through prostaglandin synthesis. And so aspirin, of course, blocks this. It's also going to stimulate the production of many liver proteins. And so we're going to get complement made. Even though complement is constitutive, it's going to increase the complement components. You're going to get alpha antitrypsin. Haptoglobin. And these are going to be things that will limit damage. You're going to get clotting factors released. Usually when you have some inflammation, there's going to be some damage that requires clotting. You're going to have C-reactive protein, CRP, and CRP binds to phospholipids on bacteria. And it acts as an opsonin or activates complement. Oops. And it activates complement via the classic pathway, which we'll talk about. Okay, so that's the acute phase response, and most of these are going to be released by our macrophages. Now let's start talking about complement. So complement are constitutive zymogens secreted by the liver. And like many inflammatory components, their secretion will increase if there is the acute phase response, triggering the liver to secrete more. They are going to function as a cascade, similar to the coagulation cascade. There are complement components 1 to complement components 9. So there are 9 complement components. We're going to have some split into 2 fragments. The small is A, and the large is B. And so, for example, you have C3 will become C3A plus C3B. C3 is a key complement component, which we'll talk about. There are 3 ways to activate, or pathways. So there's 3 activation pathways. We have our lectin pathway, which is mannan binding lectin. Or we have the classic pathway, which is IgM or IgG, plus antigen. And so this is acquired. We have the alternative pathway, and this is going to be C3 spontaneous. Now, all of them are going to trigger the same complement effectors. The difference is how they get activated, or how they activate those zymogens. What's the trigger? What is causing the activation? So let's just look at these 3 pathways as they come together. We have our classic pathway, and it's called classic because it was identified first. It's interesting it was called classic because it is actually part of the acquired immune response, in part because it needs antibodies, and in order to have antibodies you have to have the B cells activated. So we have the classic pathway, and the classic pathway is unique because it's the only pathway that uses C1. The only pathway that uses C1. Where's my C1? So C1q will actually bind to the Fc region of the antibody. So C1q binds the Fc region. And then the R and S will change conformations And this change of conformation will cause the cleavage of C2 and C4. And so they become C2A and C2B, C4A, and C4B. Now, the other pathways are the lectin, or the MBL, mannan-binding lectin pathway. And in this case, you have lectin and MASP1 and 2. Now, MASP1 is mannan-binding lectin serine protease 1. And mannan-binding lectin serine protease 1 has two, or several functions, but two main functions, is one, to activate MASP2, and two, it's a serine protease, so it can actually cleave fibrinogen and factor 13. So it's involved in the clotting cascade as well. And then MASP2 is mannan-binding lectin serine protease 2, and this is what's going to cleave complement component 2 and complement component 4. So you have the lectin, similar to that colectin. It has that globular head, and then it has that funny tail. And so you have the bacteria that will bind, and then you're going to have MASP1 and MASP2 recruited, and then MASP2 is what's going to cleave your C2 and C4 into C2A, C2B, C4A, C4B. Same thing that happened in the classic. Now the one that's a little bit different, and we're not going to go too much into the difference, but there's a few things you need to know about this one, is the alternative pathway. The alternative pathway was discovered obviously after the classic, so it was just called alternative. C3 can spontaneously break down into C3A and C3B, and it does that all the time in your body. It doesn't cause any problems because you have some inactivators that are also present in your body, and they quickly can neutralize a spontaneous C3A, C3B, as long as there's no pathogen present. If a pathogen is present, then the amount of C3A and C3B overcomes this natural ability to inactivate them, and you start to get inflammation, which if you have an infection, that's what you want. So C3B is known as the alternative, but it's also given another name, is the amplification. And we'll talk about why that is in just a moment. But so C3 can spontaneous in an aqueous environment, which our blood is, become C3A and C3B. The other way you get C3, actually let me use a different color for the C3. The other way you get C3 split is spontaneous, or you have the C3 convertase. C3 convertase is C4B plus C2B together. They can bind to many C3, and they can convert them to C3A and C3B. C3B is then going to form another complex with C4B and C2B, and it's going to be the C5 convertase. And what these convertases mean is that they're just splitting C5. So the C5 convertase splits C5 into C5A and C5B, and the C3 convertase splits C3 into C3A and C3B. Nothing different. Now what's key about all these, of course, is going to be function. So we have some big functions going on. We have amphitoxins, we have opsinin, and we have our membrane attack complex. Oops. So these are the effector functions. So all of this splitting is basically coming down to these three functions. You're trying to get C3A and C5A that are very potent anaphytoxins, and you're trying to get C3B, which is a very good opsonin. It helps macrophages and neutrophils eat things. And then you also are going to get MAC, the membrane attack complex, which is made up of C5b with C6, 7, 8, and 9. And the MAC complex punches holes in the bacteria cell wall. So all three of those are very important. Now the alternative pathway is called the amplification because it starts with C3. And as you can see, every effector function of complement is downstream of C3. So if you draw a dotted line here, oops, maybe I can't. You can see that everything above that dotted line is the activation with the classic or the MBL pathway, but there is really no effector function. C4A, some people will say, is a little bit of an anaphytoxin, but not very much. It's really everything below that line that you want to get to. And so if you have any defects upstream of below the line, you're going to have defects in complement. You might still be able to have the alternative pathway as long as the defect isn't in C3, of course, also. But just keep that in mind. So if you have a defect of C1, then you're going to have problems with complement being activated by your IgM or IgG. If you have a defect of the MASP proteins, you're going to have a defect of the lectin pathway. If you have a defect of C3, you're going to have a defect of all the pathways because C3 is needed for all of them. So keep that in mind when you talk about genetic defects of the complement cascade, because you always have to go backwards and say, okay, what effector functions are being impacted by this defect? You can also have gain of function mutations, and those mutations are typically caused by a loss of function of regulatory. So if you're not able to shut down complement, then your complement will be overactive. And so that can also happen. So again, just keep in mind what the genetic fault is with respect to the complement pathway, what the effector functions are going to be affected by that, and then the end result. Are you having too much complement activation or too little complement activation? So some things to keep in mind is when you're looking for somebody who has a complement defect, you often are going to look for, you're going to measure, for example, you can measure C3. And there's a normal level of C3, for example. And if you have too little of C3, then you're thinking that complement is overactive because your C3 is getting split into C3A and C3B. If you have too much, then your complement cascade is not getting activated and you're not splitting C3 into C3A and C3B, for example. Okay, so let's just focus a little bit more on the effectors. So anaphytoxins are basically recruiters. And so these guys are going to increase vascular permeability. They're going to allow the neutrophil to get inside of the tissue where the bacteria are. They're going to allow for that diapedesis. So they're going to increase vascular permeability. And they're going to increase leukocyte adhesion to the vasculature. Similar to what a macrophage does. They also are going to be chemotactic. And so leukocytes will have receptors for C5A and C3A on their surface, and they're going to chemotaxis towards a gradient. They're going to also stimulate the phagocytosis. Plus degranulation of our newts. So how chemotaxis works, just really quickly, you have your blood, you have your skin, and we'll say we have an injury, and so you have your lovely bacteria. And in here you'll have your complement, so you'll have your, should have drew it closer together. Let me get the hint though. So your complement will diffuse. Let's do C5a, increases. So your lymphocytes will know we have to enter here. So they're in the blood. They're saying we see an aflatoxin here. And then they'll get into the tissue and they'll follow the gradient. So wherever it's highest is where they're going to end up. And that's typically where the bacteria is. The other thing that we have is we have our opsonin. So our opsonin is only C3b. Okay, and so cells such as macrophages and neutrophils are going to have complement receptors for C3b. So we have our macrophage. Oops. And that's going to enable them to engulf the bacteria better. Just as a note, IgG is an opsonin plus C-reactive protein. IgM is not an opsonin. So for the second signal, signal 2 for our B cell, this is C3D. Just remember that C3D is going to be signal 2. So signal 1 for the B cell, of course, is the antigen binding to the B cell receptor. Signal 2 is some sort of inflammatory signal that there is an actual infection, and C3D is what gives that to the B cell. C3D binds to complement receptor 2. And that's on the B cell surface. So for the removal of immune complexes, so this is on the red blood cell, has complement receptor 1. And this can bind to C3b or C4b. And so typically what happens is your complement will bind to bacteria so they can land on the bacteria's surface, such as C3b, and act as an opsonin for a macrophage or neutrophil. But oftentimes your C3b, your antibodies, they all get jumbled up into these giant immune complexes that get into your blood and get lodged into capillaries. And so red blood cells, because they go everywhere in your body, they have complement receptor 1 on their surface. So if they're traveling through the kidney and they see C3b, it'll bind to complement receptor 1. The red blood cell does not eat the complement. It stays attached to the surface, and the red blood cell will eventually get to the spleen where you have macrophages. And the macrophages will eat the immune complexes that the red blood cell drag in. So the red blood cell kind of cleans out the immune complexes from your blood. And of course, there's also the membrane attack complex. The membrane attack complex is C5b plus C6 plus C7 plus C8. And this equals a you can call it a channel. It's not quite a channel. We'll call it a structure. And then C9 punches the hole. But C9 is dependent on the complexes C5b, 6, 7, and 8, and then it can bind to them and it can penetrate the membrane of the bacterial. Okay, so let's just talk a little bit about inhibitors and deficiency. Anything that is going to cause inflammation is very important to have controls over it. Also, because these are all constitutive, it's important to have inhibitors present in case they accidentally get activated. So there are C1 inhibitors which prevent C1 QRS activation. Well, we should put C1 Q binds to the antibody, so it's really C1 RS activation of C2 and C4. So this is the classic pathway. No other pathway uses C1. There are also C3 inhibitors. These prevent C3 activation. Especially the alternative, because it's spontaneous. And so this would affect all pathways because they all converge at C3. And there are also membrane-bound inhibitors, and these prevent MAC. These basically are proteins on the surface of our cells that, if complement is activated, MAC will not punch holes in our own cells. So they prevent the formation of the C5b, 6, 7, 8, and hence 9 cannot punch a hole. Bacteria don't have these proteins, so the MAC complex is very efficient at punching holes in the bacteria. Now, deficiencies can lead to illness, of course. You have hereditary angioedema. And this is a defect of C1 inhibitors. So what that means is if you get complement activated by antibodies or C-reactive protein, C1 will just keep activating more C2 and C4 because it doesn't have, you're defective of the inhibitors. So you get overactivation of complement. Okay, and so that is the end of this lecture. Thanks.